Bismillahirrahmanirrahim. Hello and welcome back, friends. Welcome to another video lecture by Kame Biology. In this video lecture, we are going to talk about type 2 hypersensitivity. Okay. In the previous video lecture, if you remember, we talked about type 1 hypersensitivity and we talked about the hypersensitivity introduction. Okay. Now, in this video, what we are going to talk about? We are going to talk about type 2 hypersensitivity. So, let me write type 2 hypersensitivity. Okay. Now in the previous video lecture, if you guys remember, we talked that type 1 hypersensitivity involves it involves mast cells, it involves basophils, neutrophils, it involves B cells, plasma cells, it also involves IgE antibodies, it also involves some types of the chemicals, heparin, histamine, leukotriene, leukotriene B4, C4, D4, and some other chemicals. Okay, and it can also cause, it can also cause cytotoxic reaction inside anaphylactic reaction inside our body. Now, if we talked about the type 2 hypersensitivity, so it is different from type 1. In this type 2 hypersensitivity, we will fully focus on the antibodies and we will fully focus on what? We will focus on the red blood cell. Okay, now it is very, related to our red blood cells. Okay, this type 2 hypersensitivity will sometimes it also called what? It also called cytolytic or cytotoxic or cytotoxic reaction. Now, why we call it cytotoxic or cytolytic reaction? Because in this case, such cells or they can produce, during this type 2 hypersensitivity, the antibodies are produced. Such antibodies are get hypered, or we can say antibody or toxin. Such antibody are in the form of a toxin. Such toxin are get hypered and they will cause the damages of our tissue or the damages of host tissue, we can say. So they will cause the damages of the host tissue. That's why we call that cytotoxic or cytolytic, because the antibody acting here as a toxin mediator. Okay. Now, what actually happen in this case? Two antibodies are usually produced. We call that IgG and IgM. Now, these two antibodies are usually secreted against, the host cell, or we can say against the RBC. Such antibodies will produce against the surface of the tissue cells. On such surface there will be a protein present. So such antibody will actually produce against these protein which are present on the tissue cells. On the other hand, this type 2 hypersensitivity, it can stimulate a complement system and it can also stimulate what? It can also stimulate effector cells. Now, what actually happen? Let's suppose what actually happen. This IgG and IgM are produced inside the body of type 2, inside those patients who have type 2 hypersensitivity. Sometimes we have also, sometimes let's suppose IgG. So in the first case, what actually happened? This IgG and IgM are correlate with each other and they will connect it with a C1q protein. So let me write C1q proteins. Now, what is this C1q proteins? It is the proteins which are usually involved in the complement system. If you guys know about the complement system, so this C1q proteins are actually involved in that system. If you don't know about the complement system, inshallah, I will explain the complement system in much more detail, okay? So don't worry about that. So this is actually a type of protein which are involved in the complement system. Now, their Fc portion means the IgG and IgM Fc portion are connected to these proteins. Once it is connected to this protein, then they will activate what? They will activate classical pathway. So what they will activate? Classical pathway. So they will activate classical pathway. I will also explain the classical pathway in complement system video lecture. Now once this classical pathway are activated, then what actually they do? They will activate C314. Sometime this C314, after that this C314, what actually they do? This C314 will activate what? They will activate effector cells. What they will activate? They will activate the effector cells. So let me write effector cells. Effector cells, we have neutrophils, eosinophils, platelets, macrophages, monocytes, lymphocytes. So all these cells, as I talked about, these cells are what? These cells are the effector cells. And each cell, now these effector cell will what? They will cause damages. So they will damage cell membrane, and cell membrane, then it will lead to what? Cell death. Okay, they will lead what? They will lead to cell death. Sometime what actually happens? Sometime this classical pathway, sometime they will activate this C3 protein, and this C3 protein will activate the rest of the things. But sometime this classical pathway will activate another pathway, we call it lactic pathway. So let me write lactic pathway. Now such lactic pathway will cause the destruction of the cell membrane, and when the cell membrane are destroyed, so finally the cell will what? Cell will die. On the other hand, what we have this IgG antibody. First of all, this IgG and IgM antibody are activated against the tissue surface antigen. They will activate this proteins, and this protein will activate the classical pathway. Classical pathway will activate C3, or sometimes they will activate the lactic pathway. If they activate the C3, so C3 will activate the affected cells. This affected cell will cause damages of the cell membrane, and finally the cell will die. On the other hand, this lactic pathway will cause the damages cell and cell death. If only IgG antibody is secreted, this IgG antibody will directly activate what? They will directly activate this effector cells, and then the effector cell will do the rest of the job. Okay, so then they will do the rest of the job. So that's related type 2 hypersensitivity. But on the other hand, we have the blood group system is also. Blood group system is also involved in the type 2 hypersensitivity. How? Blood transfusion system is a good example of type 2 hypersensitivity. If we talked about that, how blood group is involved. So blood transfusion system, if we transport the blood, let's suppose we have a donor and we have a recipient. Donor are those person who can donate the blood. A recipient are those person who can receive the blood. During this stage, type 2 hypersensitivity will be activated. So that's very important thing. Now before going one thing more, this type 2 hypersensitivity can cause disease, or they can cause they can cause a disease. We usually call this disease what? Erythroblastosis fetalis. So let me write erythroblastosis fetalis. Now what is erythroblastosis fetalis? Before explain this disease, let's first of all try to understand blood group system. Here I will not explain the blood group system in a detail. Okay, in some other video lecture, then we will explain the blood group system in a detail. But here we will just explain 4 types of the blood group, and we will explain RH factor as well, because such blood group and RH factor is related to this hypersensitivity, type 2 hypersensitivity. So let's explain a little bit to this blood group. Blood group system, if we talked about the blood group system, so blood group system, we have 4 blood group system. Okay, and we call that ABO blood group system. What we call that? We call that ABO O blood group system. So this is ABO system. Now such ABO system was discovered by a scientist named Carol Landster. So let me write Carol Landsteiner in 1904. Okay, so it was discovered by this Carol Landster scientist. They can classify the blood group into 8. But here I will just discuss the 4 because 8 will take a lot of time. And here we are fully focused on type 2 hypersensitivity. So inshallah in the some other video lecture then we will explain the blood group in much more detail. So we have 4 blood group system. Let's suppose this is, let's suppose, blood group A. On the other hand we have, let's suppose, this is blood group B. And this is, let's suppose, blood group AB. And this is what? This is, let's suppose, blood group O. Now, why we call that blood group A? Why we call that blood group A? We call that blood group A because they have antigen. They have what? They have a kinds of antigen as I am drawing. Now, this antigen is what? This antigen is what? This antigen is antigen A. This is antigen A. On the first thing, this is antigen A. Second thing is that this blood will secrete what? They will secrete antibody B. Whenever the blood group having antibody B against the blood group B and they have glycoprotein or antigen on their surface, which we call that A, so that's why we call that blood group A. Okay. On the other hand, similarly in this case they have also a surface proteins, so let me draw with the green marker. So they have also a surface glycoproteins or antigen which we call that B. So that's why we call it blood group B. And they will produce what? They will produce antibody against B. So they will produce, they will produce antibody against A. They will produce antibody against what? Against A. Now, this antibody A will against antibody means this antibody A will against the blood group A. So that's why we call that blood group B. On the other hand, we have blood group B. Now here is questions that why they will secrete antibody A or why they will secrete antibody B. Just think. If it produce antibody A, let's suppose this blood group produce antibody A. So it means that they will kill their own cell because anti means against. If they produce antibody A, so they will kill their own cell. So that's why the design is different. They will produce what? They will produce antibody B against blood group B. And they will produce antibody A against the antibody A. Okay. Now on the other hand, we have blood group B, AB. Why we call that AB? Because they have both two types of antigen. One antigen we have B antigen, and the other we have what? We have A antigen. So they have two types of the antigen. Due to this way, they will produce no antibody. When they produce no antibody, so then this cell will be survive. If they produce antibody A or antibody B, this cell will be die. So what actually happen? Normally they will not produce any kind of the antibody. On the other hand, we have O blood group. In this case, this blood group, they don't have any antigen on their surface. If they don't have antigen on their surface, so they will produce what? They will produce two type of the antibody. One is what we have? We have two type of the antibody. So they will produce antibody A and B. So these two antibody they will produce. So let a little bit discuss about the allele and genes. In this case, all these cells, RBC, these are actually what? These are the RBCs. All these RBC cells are controlled by the genes. Genes are actually present inside each of the RBC cells. Each genes actually control the RBC, and each genes will produce what? The antibody proteins. Sometimes genes have two alleles, or sometimes they have only one allele. If we see this antibody means this blood group AB, so they have two alleles. One is A and the other is B. Okay, this is heterozygous, and the rest are, these two are what? Homozygous. Okay, now let's talk about the blood transfusion system among them. If this blood group release antibody B, so what actually happen? Such patient will not receive blood group B. If they will receive the blood group B, so this patient may be die, or this individual may be die. So they will not receive the blood group B. They will receive, blood group A and O. A and O. Because the antibody is against B, so they will not, take the B blood. On the other hand, if we talked about this B, blood group B, so they will receive the blood from, from B, and they will receive the blood from the O. They will not receive the blood from what? They will not receive the blood from the, A. When they will receive the blood from the A, so this individual may be die. And on the other hand, this AB will receive the blood from the A, from the B, and from the O. They will receive from the blood from the all. Okay? This, if we talked about this blood group O, so they will receive only the blood from the O. Why? Because in this case, they produce anti-A and anti-B. So anti-A and A, if they, let's suppose if we provided A or B, any one of them, if we provided the blood group A to this patient, so this may be die, because they are producing anti-A. If we provide blood group B, again they will die, because it also produces anti-B. Okay. On the other hand, this is, let's suppose, a receiving process. Now, if we talked about the donor process, so this, this blood will be take to what? To, AB, and this will also take to this only AB. And this B will donate their blood to this AB and to their own as well. This O will donate the blood to all. That's why we call that this blood group is universal donor. We also call this blood group O is universal donor, and this AB is universal acceptor or recipient, because they will receive the blood from all, and they will donate the blood to all. Okay, so that's a little bit phenomena about this, blood transfusion and this blood group system. On the other hand, we have other things that we usually call that RH factor. Now, that's also important whenever we discuss the type 2 hypersensitivity. In this RH factor, what we are going to explain, we are going to explain that, you also heard from this word probably, that some people have blood group A positive, some have A negative. So that's negativity and positivity. If we talked about these charges, it is possible by this RH factor. What is RH factor? RH factor is a kind of proteins that is also present in some of the blood cells. Let's suppose if this blood group A have RH positive, means, let me change the color. Let's suppose they have what? They have proteins on their surface. These blue one representing the proteins. When this blood group A have the proteins, we call that A positive. They have proteins, we call that A positive. If they don't have the protein, we call that A negative blood. Similarly, in this case, if they have the proteins, we call that RH positive. If they don't have the protein, we call that RH negative. In this case is also, and similar in this case also. Okay, so that's why we can classify this into 8 group. But I will explain 8 group in some other video lecture. Now, that's a little bit about this blood group phenomena and the blood transfusion system. Okay, now let's focus on the type 2 hypersensitivity. What actually happen in type 2 hypersensitivity? As I told you, they can cause what? Erythroblastosis fetalysis. Now, during this disease, the mother is, let's suppose, get pregnant. So the first pregnancy will be, healthy or well, the first pregnancy. But the second pregnancy will be, destroy, or we can say there will be disturbance or problems activated in the second pregnancy. Sometimes the mother will child, usually the children will child, not the mother. But in some cases the mother are die. But usually what? The child are die. Now if we talked about this blastosis, so let's suppose we have what? Let me draw the mother here. So let's suppose this is what? This is a mother. Okay, this is what? This is the mother. Okay. Now let's suppose this mother is what? This is pregnant. Pregnant. And let's suppose this is pregnancy case number 1. Let's suppose this is pregnancy case number 1. In first case, what actually happened? This mother have also their own child. So let me draw the child, or let me draw the features or fetal here. Now what actually happened in pregnancy number 1 in type 2 hypersensitivity? Let's suppose the mother is, it has RH, negative, and his baby is what? His baby is RH positive. Let's suppose this is what? This is RH positive. Then during first pregnancy, they have the B cells also. What? They have the B cell also. This B cell is producing the antibody which we call that IgG and IgM. But this antibody is also circulating inside the baby body also through the placenta. But it doesn't mean that they will cause the death of this baby. Now the baby will not die. Okay? Because why? This blood cells will not produce anti-RH positive factor. When they are not produce anti-RH positive factor, so during first pregnancy, such antibody will be not produced, because these B cells take some time for the recognition of this RH positive factor. When they will first recognize it, so on the next time they will produce anti-RH positive antibody. So on the next step they will produce anti-RH positive. So the first pregnancy, in the first place they will not produce this, because they cannot recognize what actually happened inside the mother. So they will not produce the anti. So during first delivery, no anti-RH positive antibody is produced, due to which the healthy pregnancy will occur. So healthy pregnancy will what? Occur. Due to this healthy pregnancy, the mother is healthy and the child is also healthy. But in second pregnancy, what actually happened? Let's suppose this is second pregnancy. In second pregnancy, that's very dangerous. In this case, what actually happened? In this case, this B cells is strained. If you remember, there is a gap between pregnancy 1 and pregnancy 2. A big gap. Due to this big gap, the B cell is now trained. This is trained, and this B cell will now produce anti-RH positive factor against this. So they will produce what? They will also produce anti-RH positive antibody. This anti-RH positive antibody will also circulate inside the features, and it will cause the death of these features, or we can say the baby will be die. So when the baby is died, so the second pregnancy is failed, or we can say baby is die in the second pregnancy. Sometime this mother also die. Okay, because this they will produce a kinds of the antibody against this cells, or sometimes they will produce a kinds of antibody against this blood. So the process is disturbed. Now that's why here I am recommended one things to you guys, that before marriage you guys should have to perform the blood group test. If your partner is having blood group positive and you are also positive blood group, so then the the generation will be healthy. If your partner is negative, means they are B negative, they are B negative, O negative, or they are AB negative, and let's suppose their partner is what? Their partner is positive, A positive, B positive, then the offspring or the generation produced by such partners will be, disturbed or not healthy. Sometime die, or sometime if the pregnancy occur, or even if the child is produced, so they will be abnormal, they have the disease. So that things is usually occur in our environment. Okay, so before marriage the test should be performed. This is very important. Okay, so if we perform the test, so our next generation will be saved from different kinds of the disease. So that's about the type 2 hypersensitivity. Again, there is question arise, this type 2 hypersensitivity is activated in some other individual also? Now, because in some individual we have, let's suppose, 100 of the populations. In 100 of the population we have only 20 population having the hypersensitivity type 2, and they will produce this process, means they have this kind of process. Due to this kind of process, the cytotoxic reactions will occur. This is also called cytotoxic reaction. If you see, this is the host cell. Let's suppose this is the host, so it will be killed by this antibody. Okay, so one thing that I forget to mention about the agglutination. So here is agglutination is also occur. So let me write here agglutination. Try to remove the spelling mistake by yourself. Okay, so agglutination phenomena is occur. What does agglutination means? Agglutination is that phenomena in which antibody and antigen are react with each other, and they will cause precipitation. What they cause? They cause precipitation. We call that agglutination. In this case, the antibody will attach. Let's suppose this is the blood cells of what? This is the blood cells of the baby, and these are what? These are the antibody of mother. So each antibody, when they are arranged on such RBC cells, so they will cause the destruction of the RBC cells, and they will also cause agglutination, due to which the baby will die. So second pregnancy will be disturbed. Okay, so that's all about the type 2 hypersensitivity. I hope you understand about the type 2 hypersensitivity. If there is something missing, so then write it into a comment. Inshallah, I will explain it. If you like this video, so make sure to hit the like button, share this video, and inshallah I will also explain this blood group system and complement system in much more detail in some other video lecture. But in the next video lecture, we are going to discuss this type 3 hypersensitivity in a detail. So stay tuned and continue to watch the video. Thank you so much.